Let's get started. Good evening, everyone, and good morning, Korea. And welcome to today's AKN Research Seminar Series. It's great pleasure to introduce Dr. Brian Kim. Dr. Kim is Sol and Clara Kest, Professor of Dermatology and Vice Chair for Research in Department of Dermatology at Icahn School of Medicine at Mount Sinai. He also directs the Mark Rovol Center for Neuroinflammation and Sensation and leads the Allen Discovery Center for Neuroimmune Interactions. Dr. Kim received his MD from University of Washington School of Medicine. During medical school, he was selected as a HHA-MI NIH research scholar and conducted research at NIH in Dr. Stephen Katz's lab, focusing on roles of epidermal keratinocyte in graft versus host disease. He then completed the dermatology residency postdoc training with Dr. David Artis and Master of Translational Research at Pullman School of Medicine and University of Pennsylvania. Before joining Mount Sinai, he served as assistant professor, later associate professor at Washington University School of Medicine. Dr. Kim's works cover dermatology, immunology, and neuroscience. His lab studies how immune system communicate with sensory nerves, especially in the skin, and how these interactions contribute to the inflammation and itch. Over the years, his group has made an important discovery in atopic dermatitis, chronic itch, and neuroimmune signaling, and has published high-impact journals, including Cell Nature Science, Nature Immunology, Nature Medicine, and Immunity. His works has been recognized with several major honors, including election to the American Society for Clinical Investigation, Marion Schultzberg Research Award, and lectureship from American Academy of Dermatology, and Stephen Katz International Lectureship Award from the Society for Investigative Dermatology. His research has also had a strong translational side, including the works that help the advance JAK inhibitor therapy for inflammatory skin disease and itch. He has also served as a reviewer for numerous journals and NIH and private foundation. We are very fortunate to have Dr. Kim with us today. Please join me in welcoming Dr. Kim.
Okay, thank you, Professor Choi, and 안녕하세요. All right, I'll just kind of get into it, and please interrupt me along the way. I don't think we need to be overly formal here. The title of this is Sensory Interorgan Neuroimmune Circuits. One of the areas that we've really tried to, well, honestly, we haven't tried to do anything other than try to solve what causes itching. But we've inadvertently stumbled on this notion that the sensory nervous system may be one of the most powerful systems in the body. But I'll speak to the fact that we also think that the sensory nervous system communicates, it helps organs communicate to other organs independently of even the brain. And here are some of my disclosures. We were talking earlier that we try to take the work that we publish and then convert it to commercial entities by developing therapeutics as well. So atopic dermatitis is the disease that I started. I'm a dermatologist by training, so I actually don't have a PhD. I kind of gradually fell into the science of this. And the disease that particularly when I started my postdoctoral fellowship, that I want to go after was atopic dermatitis or eczema. This is a very common condition. Most people know about it. It's an itchy, red, scaly rash, very common everywhere in Korea, in the United States, throughout the world. And itch is the central symptom. And of course, you see rashes like this in this child, but itch is the central symptom. And yet we weren't really sure how inflammation led to itch. I know that might sound a little bit funny now, but even say 10 years ago, it wasn't terribly straightforward. It's a little bit more straightforward now. And our journey in this actually came by way of immunology. And the focus of my postdoctoral work was understanding how epithelial barriers, and particularly the skin, directly links to inflammation. And the narrative has gradually evolved over time, and now it looks very straightforward, but it wasn't so straightforward. But at that time, the alarmin cytokines like IL-33 and TSLP were fairly recently discovered and known to be produced by stressed epithelial cells like keratinocytes. And we knew at that time that these alarmin cytokines rapidly mobilize what we referred to back then as Th2 cell responses, whereby adaptive antigen-specific T cells produce IL-4 and 13 and then promote the pathology that we see. But still, if you look at my schematic here, you see how you could get barrier damage. You might get production of IgE, which underlies allergy or atopy. But it really wasn't clear how this actually leads to itch. And but however, as an immunologist, we were very interested in what other mechanisms can lead to the production of these cytokines. We discovered that rare circulating basophils, and at the time, entirely unrecognized cells like innate lymphoid cells, turned out to be rich sources of these cytokines as innate sources. To this day, I'm not quite sure what the purpose of antigen specificity on T cells is for atopic dermatitis, other than for the production of IgE downstream that results in the production of antigen-specific IgE. But beyond that, it remains somewhat unclear what the purpose of this is really at the end of the day. And we do believe innate cells play an important role. But what we also came to appreciate was that what we were interested in really was how now what we know is that whenever there's a barrier breach, and I'll talk about this at the very end of my talk, but this type 2 inflammatory axis is essentially a default of barrier breach. So if you think of barrier surfaces like your upper airway, your even your lower airway, parts of your esophagus, of course your skin, these areas are all plagued by diseases of type 2 inflammation. And this is because these are surfaces that actually face the outside world, are the direct kind of first line of contact to the outside world. And when there's a barrier breach, you actually default to a type 2 inflammatory process. But what we also came to appreciate is that wherever there was barrier breach in the skin, wherever there's inflammation, you default to type 2 and you get itch. So early on, when I started my laboratory, we just asked a very simple question, which is, can canonical cytokines like IL-4 13 directly interact with sensory neurons? It was as simple as that. The answer was yes. And they employ downstream signaling pathways that might be native to lymphocytes. And this is kind of the summary of perhaps the first, I don't know, 7 years of my work from postdoc to early assistant professorship. This in parallel, so this is all just background, led to us being able to inform therapeutics like dupilumab, which is an anti-IL-4 receptor monoclonal antibody. In parallel, as this drug was in development, we made these discoveries, and then we could directly inform that drugs like this could actually be deployed directly for itch, for conditions like this one called prurigo nodularis, which is a very itchy condition. As you can see right away, it's not something that you would typically see on someone in walking down the street, but these patients are miserably itchy. And we believe that a condition like this that's rather refractory to anti-inflammatory agents, if you could take a monoclonal like dupilumab, could intervene at this neuroimmune interface and have efficacy that we otherwise could not see with other therapeutics. And so I designed a template phase 2 trial at that time that led to a direct to phase 3 clinical trial and was successful. And this ended up being the first FDA-approved treatment for this condition called prurigo nodularis. So in a matter of about 5 years, we went from, is this even a disease, to having an FDA-approved treatment. So this is actually very heartening for us that basic science is very powerful, we believe. We continue to believe so in being able to translate rapidly to new therapeutics. The story extends to more than prurigo nodularis. This is a condition called chronic pruritus of unknown origin. These are elderly individuals who develop itch late in life. Again, many were questioning whether this is even a real condition in the early days when I talked about this 10 years ago. We found anecdotally in our clinic, these patients on dupilumab can do quite well. We published this, and this led to a direct to phase 3 clinical trial, which study A read out last year and is quite encouraging. Although it did not meet its primary endpoint, it met many of its other secondary endpoints. And in study B, we are cautiously optimistic that we may actually be able to demonstrate efficacy. And this drug may also now be potentially the first for chronic pruritus of an organ. The science it is fairly straightforward in the sense that we were asking whether cytokine receptors on sensory neurons, like IL-4 receptor, apologies, and the answer was yes. But then this immediately triggered the idea that that's actually JAK signaling downstream of this. This is obvious in lymphocytes, but in the nervous system, whether this was true in the peripheral nervous system was not entirely clear at that time. And then what we could do was we could identify then in data sets published by Patrick Ernfors and Sten Lenarson at Karolinska, we could also validate our hypothesis that there's probably JAK1 signaling within these sensory neurons, particularly pruriceptive itch neurons. I was actually at Karolinska. If you see here, you actually can't readily detect much in the way of TYK2 or JAK2 or JAK3. You can really detect JAK1. I think that was due to, sequencing depth at that time, because this is kind of the early days of single cell RNA sequencing. I met with Patrick, and this is his work here, and he told me this several months ago that they had identified that TYK2 is actually expressed in sensory neurons that actually go to joints, and that they found that in the setting of inflammatory arthritis, this is a very important signaling for pain. They published this about a month ago or so, and I would point you to that work. But I was very also encouraged by his work because, as you can see, we used his data set to prove that JAK1 was in sensory neurons. But then for him to turn around and say, oh, we also found TYK2 and it's physiologically relevant, suggests to us that this kind of motif is broadly applicable, not just to itch, but other kinds of processes. But there's much more to this, but this is old work, so I won't get into this. But this allowed us to then predict that JAK1 selective inhibitors like upadacitinib, abrocitinib would have exquisite anti-itch efficacy. Not only are these drugs now approved for atopic dermatitis, but they have exquisite anti-itch efficacy. And these are some of the most efficacious drugs for itch, particularly in the setting of atopic dermatitis. And at that time, we could not get our hands on these JAK1 selective inhibitors because they were very much proprietary at that time and not FDA approved. We were actually seeking out ruxolitinib, which was approved at that time for other indications. That led to a series of circumstances in which I was able to then design a pivotal phase 2 clinical trial for topical ruxolitinib cream, which became the actually first FDA-approved JAK inhibitor in dermatology, and is also now a very effective treatment for atopic dermatitis. I say this now because next year this drug will probably sell over $1 billion a year, which is quite remarkable for a topical drug. That's very atypical, as they typically peak sales are usually around 150 million a year for a successful topical drug. But I think it speaks to the kind of the exquisite kind of quality of this drug. But having said this, we've as immunologists, I've always appreciated it. It was quite logical that cytokine blockade or JAK inhibition, but would be anti-inflammatory. But in this context of this default barrier breach that leads to type 2 inflammation and itch, we believe that a number of therapeutics are actually absolutely neuromodulatory in fashion and have truly anti-itch efficacy at the neuronal level. So that's kind of the background of kind of how we came into this field of kind of peripheral sensory neuroimmunology. But in what I've spoken to so far is that how somatosensory neurons, which innervate the apologies here, I'm having a little trouble with my mouse. These somatosensory neurons that actually innervate the skin and soft tissues harbor these itch neurons. There's no doubt about that. But what I haven't spoken much about yet are these spinal visceral afferent neurons, which actually also house their cell bodies within the dorsal ganglia, which actually go to all manner of different visceral organs, and in fact even innervate secondary lymphoid tissues. And then there's the superhighway to the brain, which is the vagus nerve. which is 80% is sensory and goes up to the NTS within the brainstem. And that's its own system that also innervates the viscera. So this is a very simple schematic of the kind of three sensory nervous systems: the somatosensory, the spinal, visceral, afferent, and then the vagal sensory nervous system. And this is really how we generally sense the inside world, and I would say in many ways how we sense different kinds of inflammation. Um, so I'm just going to highlight a little bit of work here that Masato Tamari, a former postdoc, did, and I'll come back to this model, where he actually chemically denervated the vagal sensory nervous system only, and then he left the spinal visceral afferents intact, and we looked at lung inflammation. And we used an alternaria alternata model of inflammation where he simply instills into the airway this protease allergen, and very rapidly we can read out inflammation in the airway. And one of the things that we found very consistently is whenever we chemically denervate the vagus nerve, we get enhanced inflammation, and we can quantify this flow cytometrically in the lung. In retrospect, this isn't terribly surprising because we now know that the vagus nerve is very much a homeostatic nerve, but it still remains kind of a fairly new field in terms of the way we interrogate these systems. And of course, this paper went much deeper into the mechanisms of this, and I won't get into it because this paper was published a couple of years ago now, but I'll come back to this model later. But what we found in short was that while JAK1 and cytokine signaling in the spinal sensory nervous system, somatosensory nervous system, is very much itch promoting, what we found in the vagal sensory nervous system, there's actually a homeostatic loop whereby JAK1, we think actually, when it's overstimulated, actually results in the production of CGRP beta neuropeptide, which is highly suppressive for type 2 inflammation. Okay. So that's a brief introduction to kind of how we've explored the vagus nerve. I'll come back to this later. But we were really interested in the spinal visceral afferent nervous system. So we want to move beyond itch, and how do we itch in the viscera? Obviously not. That's a clinical question. We don't itch in the viscera, but what are analogs to itch within the viscera? Is cough a form of itch? Is urgency, bowel urgency, a form of itch? Maybe. Is bladder urgency a form of itch? This is the kind of concept we want to explore. And I say to my lab all the time, it's very important to discover molecules and pathways, but what's much more important, in my view, in science is what are the motifs? What are generalizable motifs? What are evolutionarily conserved motifs? Because that's where we can think about kind of therapeutic strategies. So Zhen Wang, who's a very talented postdoctoral fellow in the lab, decided to actually now look at the bladder. The reason why we were interested in the bladder specifically is that bladder is purely innervated by that turquoise line. It does not have vagal sensory input. It's not a somatic organ, so it's not, obviously you can't itch there, or you don't get certain kinds of pain there. But there are these spinal visceral afferents that go through the DRG and through the spinal cord, and it's purely innervated by that nervous system. So we want to start in very simple reductionist fashion to interrogate this. This is actually a really beautiful model that was published a while ago, where they actually insert the cathelicidin LL37 into the bladder, and it causes inflammation in the bladder, and then converts to a hypersensitivity in the bladder. So this was actually developed by a pediatric urologist. He actually reached out to us after we uploaded the preprint. And when we do this, what was published is that these mice actually developed bladder hypersensitivity. But what Zhen did was he decided to actually do visceromotor response, where we implant electrodes in the abdominal musculature, and then we can catheterize the bladder. And as we mechanically distend the bladder, we could read out, like an electromyogram, the electrical activity of the abdominal wall, and objectively read out if there's an element of visceral irritation. So this is kind of the gold standard for that. We also did a von Frey filament over the pelvic skin, where we tap the pelvic skin and the mice exhibits aversive behavior. And then we could also measure urgency by simply monitoring urinary patterns on basically Watman paper on the ground. And so we want to read out this bladder urgency thing, and well, not just urgency, but hypersensitivity, and really nail what are the kind of neuro as well as neuroimmune mechanisms that underlie this. So, in fact, if we do this kind of von Frey filament, we find that these mice are have much more aversive responses if they're treated with the LL37. If we now read out the visceral motor responses as we pressurize the bladder, we see much more intense waveforms than we do in vehicle-treated mice. If we now look at the urinary pattern of vehicle versus LL37 treated mice, what we find is that there's, as you would expect in control mice, these mice tend to urinate at the edges, if not the corners. There's very little dribbling, so to speak, in the middle. But whereas in the mice that are LL37 treated, you see these dots in the middle indicating that there's some element of urgency. Mice, animals in general, do not like to urinate in the middle of the field. This is precisely why when you see dogs, they want to urinate at a tree, not in the middle of a field. So presumably, if they're dribbling in the middle, that's unintentional, and we can quantify these as either small voids or center voids as readouts. So this is where the story actually takes a very interesting twist. I think this is where it really Zen's work really takes off. One thing about bladder irritation or bladder pain syndrome or interstitial cystitis is that 95% are women. They're thought to have had recurrent urinary tract infections. That's thought to be the cause. Whether that's the case, we don't know. And these patients often have other comorbid pelvic pain disorders. So they'll have irritable bowel syndrome. They might have even, believe it or not, they often have endometriosis. But clinically, they're often dismissed because by definition, somatic pain is very sharp and localizing. So in other words, if you stick a needle in me, I can tell you exactly where that needle went right away and I'll feel it. Whereas when I have abdominal pain, I can't tell you often where it's coming from. It's hard. I'll say, I don't know, I feel like it might be my colon. Maybe it's my bladder. I don't know. And that results in a lot of clinical skepticism when a subjective symptom like pain. As a clinician, I'm very aware of this because my itch patients have often been dismissed. It was only when we had treatments that suddenly people believe them. So this is a journey that's very familiar. Well, I asked Zen this question when they said, maybe this comorbidity is real and we can test this very objectively in mice. So let's just look in the colon in these mice that have been irritated in bladder. So we have not assaulted the colon whatsoever, but now we do a VMR where we inflate the colon and we measure this kind of irritation. And what we found was that as you pressurize the colon, you find enhanced irritation in the colon in these mice. And then as well as what we find is that there's actually a delayed expulsion time as well if we put a bead through the intestinal tract. Now, why is that happening? So this goes back to the sensory nervous system. So the interesting thing about the sensory nervous system is that if you stick a needle in my big toe, that neuron actually has axons that go all the way to almost to my spinal cord in my lower back. And that's where the cell bodies are. That's how long they are. So we hypothesized, and honestly, people had hypothesized this for decades, and there were tracing studies that were done to suggest that this possibility for sure. But we wanted to look at this very rigorously. For one, we really felt this is possible, that if you can have axons that are that long, why can't you have axons from one nerve, one cell body to multiple organs in the pelvic viscera? So that was our hypothesis, that you could actually have one neuron that innervates multiple organs. So to test this, what Zen did was took chlorotoxin B conjugated to two different fluorophores. Chlorotoxin is a great neurotoxin. It enters and it gets retrograde transported to the cell bodies, which you're looking at here in the dotted lines. When you pull out the ganglia, you can see these cell bodies. And what you find is that the respective organs have fluorophores that now trace back to the DRG, the dorsal root ganglia, and you find that you see there are neurons that strongly suggest that they are from both organs now, because we have these fluorophores converging. You see this washout in gray or white. And these have been referred to as convergent dichotomous neurons in the past. We refer to them as polyorganic sensory neurons. And we, in collaboration with Juan at University of Michigan, decided to actually look at this much more deeply. So what this data is essentially in vivo calcium imaging. So we're looking at calcium reporter at the cell bodies, and then what we're doing here is we're distending the bladder, and then we're distending the colon. And you find very clearly here with bladder distension, you see activation of individual neurons by calcium response, and you see enhanced activity as you get from as you pressurize the bladder. Down here, we find that these are colon responsive neurons. So as you distend the colon, you get responses of neurons within the DRG here. Interesting thing is if you look above this dotted line, you find that there are neurons that are responsive from the bladder, as well as at higher pressures in the colon as well. At steady state, these are the polyorganic sensory neurons, we believe. Now, if you go into the LL37, once the mice have gotten inflammation in the bladder and irritated, what happens is you see more neurons that are dual responsive between the bladder and the colon. So we believe these studies strongly suggest that there's actually these dual responsive neurons are highly functional, and they actually acquire this kind of sensitivity upon bladder insult. And this actually goes to both the bladder and the colon. And I'll come back to this concept in a little bit. But we wanted to, again, these neurons have been described. There was incredible skepticism about what they exist. In fact, I would argue they were largely dismissed. So we wanted to take the more rigorous approaches yet again. We took an intersectional genetic approach. So now we have mice, AI224 mice from the Allen Institute. We collaborated directly with the Allen Institute, where they now have flipocytes, flanking stop codons, as well as LOXP, upstream of EGFP and TD tomato, respectively. So now what we can do is inject AAV packaged with pre recombinase into the colon, and then AAV packaged with lipase into the bladder. Intersectionally, what will happen is these will now retrograde transport to the cell bodies, which you're looking at here. If they are from the colon, they're green. If they're from the bladder, they're red. If they're dual, they should be yellow. So you see green, red. Most of them don't overlap, but you do see a corresponding yellow neurons that we believe are these polyorganic sensory neurons. All right, so we already did this with the CTB. Why are we doing this with AAV? Well, it's an orthogonal approach, but also now we can get much more sophisticated with it. We can now use the EGFP and the TD tomato as barcodes as well. And now we can actually induce spatial transcriptomics. So when we do spatial transcriptomics, what we find is we will find occasionally, or approximately around 10% of the time, we'll find these neurons that are clearly dual positive for TD tomato and EGFP. Again, we believe these are the polyorganic sensory neurons. So the nice thing about spatial transcriptomics, you could take a probe set of 500 markers, and then you can really subset out which neurons these actually are. And when we do this, what we find out is that we can then map the probe set onto existing single cell RNA sequencing data sets. These were data sets from recent data sets from Patrick Ehrenforth, who I mentioned earlier, and David Ginty, who both actually won the Brain Prize a couple months ago. And when we map this back, what we find is that we can identify that these dual labeled neurons that are EGFP TD tomato positive are largely the CGRP gamma 2 subset that was originally described by David Ginty's group. What's interesting about this subset is positive for a number of different markers shown here, but in particular CHRNA2. Why is this important? Well, the CHRNA3 positive neurons were reported by Stefan Leichner's group in 2017, and what he termed silent nociceptors. These are nociceptors that are normally fairly, they're just normal nociceptors typically, but what happens is following inflammatory insult, they acquire mechanosensitivity. So this is exactly what we would expect in the bladder, where a normal bladder, you get distension, no irritation. But in the setting of interstitial cystitis, because there was some kind of severe inflammatory assault, those nociceptors have now become mechanosensitive. So any kind of mechanical stimulation of the bladder above a certain threshold causes a lot of irritation, which is exactly what these patients experience. The difference here is we believe that these are actually polyorganic neurons that have this kind of identity. We took another intersectional genetic approach using AAV the same way, but this time in a different mouse, where we actually wanted to telescope in on these polyorganic sensory neurons. But this time what we're doing is we're inserting GI dread to insert an inhibitory G protein coupled receptor only in these polyorganic sensory neurons. So then what we could do is we could give clozapine to these mice, otherwise inert compound that activates an inhibitory GPCR. And then when we do this, what we find is that we cannot get rid of the bladder hypersensitivity because the bladder, many neurons are activated in the bladder, but the only neurons that convert and transmit this hypersensitivity to the colon would be the polyorganic sensory neurons, which would be inhibited by the clozapine. And we find that we get attenuation of the colon hypersensitivity as well as the motility issue defect when we do that. So we feel that this is just proof of concept. These neurons can potentially be targeted. Bringing this to a bit of immunology, if we now look at TRPV1 nociceptor reporter mice in red and then avidin staining in green, what we find is sheets of beautiful mast cells as well as beautiful TRPV1 positive neurons. When you look in the superficial lamina propria, what we find is that the vast majority of these mast cells are in close proximity to these TRPV1 neurons, while when you look deeper into the bladder, in the muscularis propria, we see that these mast cells are somewhat agnostic. Why is this important? Well, mast cells have been implicated as potential culprits in interstitial cystitis, bladder pain syndrome. This has been something that has been considered for a long time. But also, we had worked with Xinzong Dong, who discovered MRGPRs, the whole entire family of MRGPRs, but importantly, MRGPRX2, which is on human mast cells, as well as MRGPRB2 on murine mast cells. And we found that this is an alternative activation pathway for mast cells that's very important for itch. This is now back to itch in the skin. And then we suspected that could this process be important in the bladder as a triggering event upstream of these polyorganic sensory neurons. We have MRGPRX2 transgenic mice in which we've deleted the B2 endogenous murine B2 and inserted the human X2, and we find that they're only expressed in mast cells. Why is this important? This is important because in order to pharmacologically test compounds like EP002, which is one of the first MRGPRX2 antagonists, you need human X2 in the mouse. It does not target MRGPRB2, the native mouse receptor. When we do this now, because it's upstream of the polyorganic neurons, what we find is we can attenuate both bladder and colon hypersensitivity and reverse the motility defect. So we believe that this is a really exciting work by Zen, because really we're fundamentally, I think, determining that these sensory neurons are yet important again in a very different fashion. They're not just important for itch, they're not just important for lung inflammation, but they're also very important for communicating signals, nociceptive signals from one organ to the other. And we believe that this is an evolutionary conserved homeostatic kind of wiring within, particularly in the viscera for coordination, and to preserve kind of coordination in the periphery in a way to unburden the brain from having to process every kind of coordinating signal. But then, of course, in the setting of pathology, this could cause a lot of miswiring and misperception and cross-organ irritations, a concept that's been well appreciated clinically, but the mechanisms have been fairly elusive. Okay, so then I just want to end in this last part with another story by Rintaro Shibuya, who's a very talented postdoctoral fellow who just left the lab. This work, it was provisionally accepted recently But I think is a very interesting piece. So if you actually look now back at the vagal sensory nervous system, again, the superhighway to the brain from the viscera, there's one branch that actually does not go to the viscera. There's the auricular branch of the vagus nerve that actually goes to the ear. The interesting thing about this branch is that it probably does not go to the same ganglia as the other. It probably goes to the jugular ganglia, and these go to the nodus ganglia. But they're somewhat separate in humans, but quite in close proximity in mice. But nonetheless, we think that they're different ganglia. But for some reason, this nerve is thought to be able to manipulate a whole host of visceral processes. And in fact, there are a number of companies that have developed vagus nerve stimulators transcutaneously through this auricular branch that are, very serious companies going for interesting indications like rheumatoid arthritis or long COVID or even depression, anxiety, a whole host of different conditions. And this is kind of a hot topic. So we wanted to explore this much more in more extreme fashion. In other words, we wanted to test, is this a real thing? Is this testable in binary fashion? Can we determine, is there an importance of this auricular branch of the vagus nerve? Well, we know, this is tracing studies that Masato did early on. We know that really the vast majority of the airway is actually vagal ganglion neurons shown here on the left. So if we actually inject AAV Cree into the airway and trace back in stop flox reporter mice, very few actually go to the segmental dorsal ganglia. It's really all vagal sensory ganglia. So in contrast to the bladder, which was a pure DRG organ, we wanted to go to a very pure vagal organ, which is the airway, to test this idea that this transcutaneous vagus nerve may be actually important in inflammatory modulation. So what Rintaro did here was very interesting. He actually injected resiniferotoxin, which long-term will chemically denervate the nerve, and it does so by hyperactivation of TRPV1, and then it causes chemical denervation. In the short term, when you're looking at in the first week, it just simply, you're only seeing the hyperactivation of this nerve. You're not seeing the denervation. And we ran this through the alternary alternata model that I described earlier that Masato had set up in the lab. And what actually Rintaro found was that when you hyperactivate the nerve in near term, this activated nerve actually suppresses the lung inflammation, ILC2s, and eosinophils. And you see that, in fact, compared to controls, that there is improved pathology. And then, of course, cytokine levels are also reduced, particularly IL-5 type 2 cytokines in this context. Of course, this is slightly, one could say, oh, well, RTX could be leaking all over the mouse. How do you know that's the auricular branch? So we here decided to take a slightly different approach here, whereby we now we took TRPV1 Cree mice, crossed them to actually the now GQ dread mice, in which we can, by way of TRPV1 Cree, insert the activating G protein coupled receptor, and then we could locally inject clozapine here into the ear, and we get this similar immune suppressive effect in the airway with local injection of the clozapine in these mice. Now but one could argue here, well, you've inserted it into every nociceptive nerve. If the clozapine leaks throughout the mouse, who knows what's really happening, right? So we took a slightly different approach. What we did here was we used AAV again and inserted the GQ dread directly into the vagal ganglia, which only gives one branch to the skin, which is the ear skin. And then we did a local injection of clozapine here. So now the only nerve that we could possibly activate with a clozapine is vagal derived only. We're not going to be able to activate any other nerve with the clozapine. When we did this, we also saw suppression of the inflammatory inflammatory process in the lung. Linking back to Masato, our other postdocs work, we found that when we do this, the CGRP beta levels actually go up, which, as I mentioned earlier, was an immunosuppressive effector neuropeptide. Well, what if we do the reverse? Now, what if we insert GI dread, inhibitory dread, directly into the vagal ganglia, and then now inject clozapine? Well, what happens here? We get actually now the opposite effect. We actually aggravate inflammation in this context at the cellular, histologic, and a cytokine level as well, and we get now loss of CGRP beta. Okay. Okay, so now one might say, okay, I don't know, maybe your AAV is leaky, all this stuff. So what we did here was something slightly different. We used AAV and we inserted channel rhodopsin into here. So now what we could do is just stimulate with blue light to activate channel rhodopsin in the auricular branch of the vagus nerve. And when we did this, the blue light alone was enough to actually suppress the inflammation in the airway with all the same parameters as I showed here. We get a bump in the CGRP beta as well. So, what we think is actually that this transcutaneous vagus nerve may be actually quite important in regulating this. The exact mechanisms are less clear. We're actually thinking about actually exploring this a little deeper. But hopefully what I've highlighted to you is that, there are many sensory nervous systems throughout the body. It's important for itch, but it's probably important for many other processes as well, such as visceral pain. And there are many ways in which we can actually explore inter-organ communication to really mobilize these different effects. A lot of this work was made possible by the Allen Family Philanthropies, which funded a consortium for our center to build out, combining neuroscientists and immunologists to kind of put together some ambitious ideas. So I have to really credit them. We were funded by them a little over 2 years ago, and a lot of this work happened subsequently from that as well. But I also want to end with the idea that I said earlier, that motifs are more important than molecules and pathways. What we want to quickly do is zoom out and figure out what are the motifs, what are some takeaway lessons. So, I study allergy a lot, and one of the things that we're appreciating is that I mentioned earlier that barrier surfaces that face the outside world default to type 2 inflammation. They may default to symptoms like itch and cough and all that. Well, you have the upper airway, you have the eyes, you have the mouth, you have even the lower airway. These are areas that are largely plagued by type 2 inflammation, whether it's allergic rhinitis, eosinophilic esophagitis, asthma, even food allergy in the stomach. And all of these, from the belly button up, are largely the symptomology is often mediated by the vagus nerve. We refer to this as allergy, and we have IgE responses. And in fact, blocking IgE is a therapeutic for many of the conditions I listed. But once you get below the belly button, it's no longer the vagus nerve. It's much more the DRG, the spinal visceral afferents. It is no longer allergy, it's irritation. It's no longer IgE, it's much more MRGPRX2. And I think that this field of neuroimmunology helps us to understand these broad macro kind of metabiologic motifs that it's very hard to understand if you're only studying allergy or if you're only studying somatosensation. It's really if you zoom out that you're able to observe these motifs. And I think they do help guide us to better ideas about how to chart biology in new ways, but also develop new therapeutics. So, it started with what I thought was at that time, I thought it was an ambitious idea that to actually find out what causes itch. But in retrospect, that was actually a rather modest proposition. There's a much bigger world that we're uncovering about kind of sensation throughout the body and regulation of inflammation by way of the nervous system. So I just want to end there. I want to highlight in particular early work by Landon, and some work by Masato, Zen on the bladder, Rentaro with the lung. All of our funding agencies, our wonderful collaborators at the Allen Institute in particular that really helped us, and Bo Dwan at University of Michigan. So I'm happy to answer any questions in the remaining time. Thank you all for having me. I hope it's interesting. I know some of us on the East Coast are up late, so thank you.
Thank you. So we are open for questions, so please raise hand and then feel free to ask.